small local NGOs to big organizations like the World Economic Forum. So it's clear that we will dedicate a large amount of resources, financial resources, human resources, to the restoration of native ecosystems. But we still lack most of the knowledge bases necessary for doing it right. So what I'll show you in this presentation is how I have addressed some of these knowledge gaps in collaboration with partners in different regions of Brazil and in other countries. So it's critically important to decide why to restore an area, where the restoration interventions will be implemented, and how they will be implemented, because the final outcome rely on the intersection among these three questions. And most of the times, people is not aware of this, of the full complexity of ecosystem restoration. Many of them believe that it's a simple act of digging a hole, putting a tree seedling there, and just leave it grow. And but this is not the reality. We do need high quality science for achieving the best outcomes from this activity, as well to avoid unintended negative consequences. So regarding why to restore, we have to acknowledge that restoration can provide many benefits, like climate change mitigation, food security, water purification, biodiversity conservation, and local livelihoods. But difficultly, all of these benefits can be maximized in a single site or in a single project. And we have to make decisions. Which are the priorities? And these decisions are important because they may reflect some power asymmetries. So for instance, developed countries, they may want to invest in restoration to mitigate climate change, but local farmers in Brazil, they don't care about climate change. They care about water, they care about wood, they care about food production. And then it's challenging to balance and to harmonize the disputing values and objectives when we have a restoration project. So I have also worked at the governance side of restoration in order to find ways to reduce trade-offs and maximize synergies and harmonize the relationship between among different stakeholders involved in the restoration process. And this is critical because restoration can cause problems if not well done. So there are, massive project failures resulted from the lack of maintenance of or poor planning. So sometimes people, they plant trees to improve water production, but the outcome can be the opposite because trees, they have a very high leaf area surface and they evapotranspirate a lot of water. So tree planting can, in fact, reduce water yield at the first years of the process. At the same time, people sometimes they plant trees for mitigating climate change, but they can do it in ancient grasslands, high diversity and highly threatened grasslands communities like in Africa. It's the case, some tree planting programs are destroying native and important ecosystems, or they can use directly destroy native ecosystems. We may also displace farming activities and contribute to food insecurity. So there are some major problems that can be caused by restoration, and we have to avoid it using good science to help decision makers to plan restoration accordingly, which can in fact provide many potential benefits, like some of the ones I mentioned before. So regarding where to restore, I'd like to show here a multi-scale collection of studies that highlights how we could improve restoration outcomes by deciding where they should be implemented. So I'll start with a research project of one of my students. So we worked in the watershed that supplies drinking water to my city. So we used past land use maps to create a map of regeneration probability. So we understood which factors drive the chances of forest regeneration, considering, for instance, the distance from remnants, from water courses, slope, previous land uses. So we create a map of restoration probability. We investigated the costs associated to different restoration interventions. And then finally, we created a spatially explicit map of restoration costs, which have been used by local governments to decide where to restore to maximize the budget they have for this activity. Moving to another scale, the Atlantic forest scale, the biome where I live, it's one of the global hotspots for biodiversity conservation. So under the leadership of my colleague Bernardo Strasbourg, we created, using linear programming, we created a spatial prioritization map of restoration in the Atlantic forest, which is able to triple conservation gains in half the costs. So we can have, like using this example, a massive gain of restoration, feasibility, and benefits depending on where restoration is done. And then moving now to the global tropics. So I led a work on restoration prioritization for tropical rainforests and found which areas in the world could be considered like restoration hotspots, areas that can maximize the benefits. So we considered here biodiversity conservation, climate change mitigation and adaptation, and water security, as well can maximize the feasibility of restoration. We consider land opportunity costs and the persistence of forests over time, and also the chances of biodiversity, the risks of biodiversity failure in these projects. And finally, there is a new work led by Bernardo, which will be published in 2 weeks, that identified, using the same approach as I described here, global opportunities for all terrestrial ecosystems in the world. So you may soon check this new research that identified the global opportunities for restoration considering all terrestrial ecosystems. But another critical issue is how to restore. So even if we do restoration in a very potential area, the outcomes can be lousy depending on where you do restoration. And there are many different ways to restore an ecosystem. And here I bring the examples of forest ecosystems, which is a kind of ecosystem that I work. So although ecologists, they may prefer, mixed native species plantings or natural regeneration, local farmers, they may prefer agroforestry systems and other strategies to restore a forest that can result in some income for them. So when decide how to restore, we have to integrate both ecological and social issues to decide the best solution for each condition. So I have also worked with the development of, restoration systems that could be better applied to different socio-ecological contexts. Moving from tree plantings, as you can see here in the left side, from the use of forest topsoil, the organic matter and soil and seed banks from forests that will be legally degraded and cleared, and then integrating exotic profitable species with native species for allowing some income to farmers. And then finally, like Barbara, I'm also working with LiDAR technology. So I'm working with a drone equipped with a LiDAR and hyperspectral sensors. And we are using it to monitor some of these restoration projects at large scale, and then integrating the LiDAR data we have with radar and high resolution satellite image, as already shown by Barbara yesterday. So we have been able to track restoration development using multiple indicators.
40 seconds.
Okay, so we are also using some of this technology to support decisions at the policy level. So thank you.
I was not the reason to stop you, but thank you very much for your excellent report on ecological ecosystem restoration. We're now moving to Dmitry Vladimirov, Russia. 12 minutes. Go. I think we're experiencing sound problems. Dmitry?
Yep.
Okay. Now we can hear you. Go ahead.
Are we good? Are we good? Can you see
Yes.
my PPT?
Yes, we do.
Okay. So, honorable colleagues, the study of plant invasions is the science that attempts to understand causes and consequences of plant introductions outside their native areas. Invasive plants have an impact on global biodiversity and ecosystem functions, and their management is complex and formidable task. The applied aspects of this study include the health and economic impacts of invasions. Before I'll start to present the results of my research, I would like to focus on some theoretical aspects of the study of plant invasions. Let me try to do. Okay, so first of all, how do we understand plant invasions in Russia? Because this is a very complicated topic, and there are lots of different opinions how to understand this in Europe, in the US, and Brazil. And so in Russia, we understand plant invasions are all cases of plant species introduction to ecosystems outside their native areas and settlements in new places to such a degree when they cause economic or environmental harm and or harm to human health. We call the list of these species the blacklist or the invasive fraction of flora. Well, this definition is very close to how do our European colleagues from Central Europe understand plant invasions. And here below, I would like to show two examples of biological invasions that I saw. One in Russia. It's Ambrosia artemisifolia, very familiar species for one who are from or maybe been to the North America, and Humulus lupulus. It's one of the most aggressive invasive species in China. And any invasive plants or invasive pallium plants, so there are different specialists call them indifferent, and they are heterogeneous by origin and heterochronic by the resident status and naturalization, a group of plants in the regional flora. Or not only regional, but I'm as a regional scientist, I'm talking about certain regions. This group develops as a result of transcontinental, transzonal, and interzonal migrations of species, and they are carried out because of direct or indirect human activities. The alien invasive species, or simply invasive species, are usually characterized by a rapid distribution and ability to master a wide range of natural and anthropogenic ecosystems with the native plants displacement. And three more examples of invasive species from Russia and from Voronezh region. First of them is Acer nigundo, an American species. It's an example of transzonal migration. Impatiens glandulifera, it's the species from Himalaya area, and I think that Mr. Dad, who presented his PPT yesterday, might recognize this species. And Tribulus terrestris, this is the species of Euro-Asian steppes, and it's an example of interzonal migrations. We divide all invasive species using three basic classifications. Let me say just a few more words about third classification. According to that, all invasive plants are distributed among four invasive statuses. Invasive statuses, this is a very common characteristic, and first I found it in papers of American scientists, and we changed them a little bit, and I just want to specifically stop on the first one. So the transformers, or in Russia we call them edificators. So the species, they do not only naturalize in natural and semi-natural ecosystems, but also destroy existing ecological succession connections. They usually act as ecosystem engineers and dominants from form monodominantal communities and displace native species. I think this group is very familiar to all ecologists who had deal with invasive species. And I can give some more examples of species like this from each country. It's Alternanthera phylloxeroides from China, or Lantana camara from India, Acacia abunce or Chondrilla unice from South Africa, and Arunda donax from Brazil. So this is a very common problem. In general, it is called homogenization of biota. I won't stop on the other three statuses. They are not so aggressive, but the status 2 is also might be very aggressive, especially in Voronezh region. Well, and Voronezh region, what can I tell about this one? In the it's a territory in the European part of Russia, and I gave general information about Voronezh region here. The climate is temperate, and what's important, the territory of Voronezh region is not very big. It's like Switzerland size, but it lies on two natural zones. It's steppe and forest steppe. And just in these three photos, I had some natural ecosystems of Voronezh region. This is originally how did Voronezh region look like before the middle of 20th century, and some relic landscapes like calcited steppe flora and the Don River basin. And now I would like to say some information about research results in Voronezh region. Well, in European part of Russia, there are about 277 invasive species. The most widespread invasive species are Acerigunda, Echinocystis labata, Eladea canadensis, and Erigeron canadensis. All of them, except Echinocystis labata, are from America. And in Voronezh region, the blacklist includes 120 vascular plants. When I prepared my PhD thesis, I prepared also 120 maps of their spatial distribution. Now this information this information is spreading, and I have more information about their distribution, because invasive species, they are very mobile and active, and they can change their invasive status in time. We also made analysis about invasive species taxonomic spectre, and leader in this spectre is Asteraceae family. The second is Poaceae family, Rosaceae family, Brassicaceae, and the last one is Henapodiaceae family. Among plant life forms lead herbical monocarpics. That means that invasive species, they are still their invasive process is proceeding, and they are still cannot find the best ecological niches. And among geographical groups, leader is America, North America to be certain. And 50% of all invasive species migrated in Voronezh region due to indirect human activities. This is important because usually invasive species migrate indirect human activities. People introduce them in botanical gardens. They use them. They use them in green buildings, and so on. And all of these species were divided into 4 invasive statuses. The most important one and critical one is the first one. So you can see that 7 species now have the first status. And the fourth, I think one of the most important issues and of our work is that we identified and classified the main corridors of invasive species migration. They are river valleys and main transport roads like autoroads and railway roads. This is not in use, and our colleagues from Europe and China and India, they have got the same results. And I think it's very important, especially when I will talk about modern corridors like New Silk Way and corridor north-south. And here are 2 examples of maps that I prepared. Just short information about the species. It's Ambrosia artemisifolia, and the map which shows us its spatial distribution within the region. The letters inside yellow parts of a map. This is acronyms of herbariums where we found the first herbarium
1
samples.
minute.
Okay, and briefly would like to share with you information of our cooperation with the Chinese colleagues. Sorry. We have been cooperating with Chinese Sichuan Provincial Institute of Natural Resources Sciences since 2014. During that time, we did a great job. We clarified what are the invasive species in Chengdu city. We prepared maps. And now we also publish some papers in Russian and in Chinese. For a moment, in process is alien invasive plants in Chengdu. And then one last thing. So corridors of migrations, possible territories for future joint scientific work of China, India, Brazil, and South Africa. First, it's the international north-south transport corridor. Of course, invasive species will use this corridor for migration. Another is the New Silk Road, and we have already started to do some research with Chinese colleagues to identify invasive species which are using this corridor for migration. And the third one is major seaports of BRICS countries. These ports could be places of concentration of invasive species, and then after they concentrate them, they can migrate along railway roads or autoroads. Thank you for your attention. We'll be happy to answer your questions.
All right, thank you very much, Dmitry. Please stick with the timing. I encourage everyone to do so. And also, I remind you that you could just send the questions for the speakers in the group chat. We're now moving to Swati Singh from India. Swati, are you there?
Hello.
12 minutes, go.
I cannot share the slides because Dimitri has not stopped sharing the slides.
Sorry, yep. Okay.
Okay. So, hello everyone. I'm Swati and I'm a PhD student. I will be presenting my work on recycling of industrial waste for the degradation of chlorinated organics. This work was done under the supervision of Professor Anurag Garg at Indian Institute of Technology, Bombay, India. Since my work is slightly different from what everyone else has been presenting up till now, I would request you to take a moment and read the following newspaper headlines. As you can see, they're all pointing towards water pollution in our rivers, which is for developing countries. There are many sources of water pollution as shown in the figure, but I would like to highlight 3 anthropogenic sources. First is the agricultural runoff of fertilizers and pesticides. Second is the rapid urbanization resulting in increased sewage, pharmaceutical compounds, and personal care products in our water. And third is the industries which contain toxic metals and non-biodegradable organics. Also, solid waste can contribute towards water pollution, as in developing nations, the waste is generally dumped into the grounds, and a variety of waste reaches the rivers and oceans. And other than that, there is leachate which is produced in the dumping ground, which can contain water. Now, thermal power plants, pulp and paper mill, textile industry, chemical manufacturing industry, they are all water-intensive industry, which means that they require large volume of water for the production process. This table in the slide shows the volume of wastewater generated by various industries into River Ganga. River Ganga originates from Himalaya and eventually ends into Bay of Bengal. And what we can see is that pulp and paper mills were one of the biggest contributor towards River Ganga pollution. Now, the process of paper making can be simplified in these 4 steps. The first step is the raw material preparation, which involves debarking and shredding of the wood. The second step is pulping of the wood chips, where the wood chips are fed into a digester with some chemicals so that it is converted into pulp. After the pulp is produced, the pulp is bleached so that we can get the white paper. And this is done in a series of bleaching steps, bleaching sequences. And the last step is when the pulp is converted into paper. Now, during these steps, there are a variety of pollutants which are produced, such as organic pollutants, color imparting substances, inorganic compounds, and chlorinated compounds, and suspended matter also. Now, amongst all these traditionally, chlorine was used as bleaching agent in paper mill, which resulted in the production of chlorinated organics. They are collectively called as adsorbable organic halides, or AOX. The use of chlorine has been phased out in developed nations, but due to economic consideration, developing nations like India are still using chlorine-based chemicals for bleaching. Chlorophenol are one of the constituents of AOX, and they are recalcitrant. They possess health risks to humans, and they have their toxicity increases with increase in chlorination. So the discharge of these organics into our rivers is a major concern to the water quality of the rivers. So chlorophenols are basically substituted phenols with 1, 2, 3 or maximum 5 number of chlorine atoms, and as I already mentioned, their toxicity increases with increase in the chlorine functional group. So pentachlorophenol will have the highest toxicity, actually. So in my study, I used 4 of these compounds. I used a mixture of these compounds, and I'll move to my study. I'll move to my work. So we chose a process which is called photofenton oxidation. We chose this process because there was a good literature available on it, and it was quite efficient in degrading recalcitrant compounds. Now, in order to ferrous sulfate as catalyst, and we added hydrogen peroxide as oxidant. This mixture was fed into a UV reactor. The orange thing in the reactor indicates that there's a UV lamp in the center, and the solution was continuously stirred to maintain the homogeneity. Based on the experiments, we drew some graphs basically from design expert software. So these are response surface plots. They are typically used to determine the optimum conditions. Now, design expert can also be used to design experiment, and it gives you the effect of individual variable and interaction of variable, and through this we can predict the optimum conditions for a particular set of experiments. So the process was found to be very efficient, and the optimum conditions were also obtained. Now, this slide shows the simplified mechanism of photofenton process. So I will not go into much detail. I just want to highlight the first reaction, which is when you have iron sulfate or iron in the form of ferrous iron, and you have an oxidant, it converts into Fe3+. So there is an oxidation, and at the same time there's a reduction. So it's a mixture of redox reaction, and during this it generates OH radicals. Now, OH radicals are very reactive species, and they are the ones which help in the degradation of recalcitrant compounds. So I'll move on to the next slide. Now, despite the high efficiency of this process was limited due to high cost of chemicals and UV lamps. Now, in this regard, the development of a low-cost catalyst would, from a waste material, will reduce the overall cost of the process. So therefore, our next objective was to explore waste which contains iron so that we can replace ferrous sulfate. So we started with steel industry waste, and we did a meta-analysis of steel industry waste, and we found out that it contains about 30% of iron, like 300 milligrams per gram. So this waste was modified to produce a photofinton catalyst. Now, in a heterogeneous catalyst, the oxidation reactions, they take place on the surface of the catalyst. And also, if the ions, like iron, is leaching from the catalyst, there can be homogeneous reactions as well. And this process is usually able to produce OH radical, and it can react with your original compound to produce carbon dioxide and water. Now, this is the X-ray plot of the catalyst that we prepared. Now, what we found was that that iron was present in the form of iron oxide, and mainly magnetite and hematite, which facilitate the surface redox reactions. Now, this is another set of results in which we did the same experiment in the absence of UV light. So we wanted to see what is the effect of presence of UV and in the absence of UV, because UV lamps add to the cost of the process. So that process is called Fenton-like process, and we saw that in case of our bleaching wastewater, the difference was not very high, but in case of our pure compound, there was a significant difference was obtained for a particular catalyst. So other than this, we also performed adsorption control experiments. So whenever you're using a heterogeneous catalyst, you are expected to perform an adsorption control experiment, and the removal due to adsorption was found to be less than 15% in our study. So we concluded that our waste contained high iron, about 40 to 50%. Our waste contained about 30% iron. Our catalyst contained about 40 to 50% iron, and this iron was present in magnetite and hematite form, and the process could remove about 64% of organic carbon. And we also tried to see whether radicals are actually produced or it is just an adsorption experiment. So we also carried out some radical scavenging reaction, and we found out that radicals were involved in the oxidation reaction. And lastly, the advantage of this catalyst was that the iron leaching from the catalyst was less than 2%. So additionally, I have worked with I have also explored other waste materials such as activated sludge from sewage treatment plant, black liquor from pulp and paper mill, and press mud from sugar industry. So these were converted into activated carbon, and then they were used as catalyst support material, and iron was impregnated on these substances, and then they were used as catalysts for the degradation of chlorophenols and for the degradation of stimulated bleaching effluent from pulp and paper mill. And these are the papers that we published from the two studies. That will be all from my side. So thank you so much for listening to me. If you have any questions, please feel free to ask.
Thank you, Swadis. Thank you very much for your excellent report. I appreciate the report on the recycling of the metallurgy waste. Great example. Well, we're now moving to Kungfeng Q, China. Hello.
Hello.
12 minutes, go.
Okay, thank you. Dear everyone, it's my good honor to attend the 5th BRICS Young Scientist Forum. I'm Kungfeng Qiu from the China University of Geoscience in Beijing. The topic I would like to share with you today is global antimony and the environmental considerations. Antimony is an important mineral commodity used widely in modern industrial societies. Here are the outlines. Identified resources of antimony are unevenly distributed around the world. This figure shows the locations of selected antimony deposits, mines, and major occurrences. China has the bulk of the world's identified resources. And antimony is an important mineral commodity used widely in modern industrialized societies. It is used in lead-acid storage batteries for backup power and transportation, in chemicals, ceramics, and glasses, in flame retardant materials, and in heat stabilizer and plastics. Graph showing world production, US apparent consumption, and US mine production of antimony from 1900 to 2012. In 2013, China produced about 80% of the world's supply of antimony. Its leading role in antimony production is greater than it is for any other minor metal. And the geology part. Antimony is a chalcophile element, meaning that it combines readily with sulfur. It is found in more than 100 minerals, and stibnite is the most common antimony ore mineral. Antimony occurs in a variety of deposits of various ages, including epithermal veins, pegmatites, and replacement and hot spring deposits. Simple coarse stibnite vein and replacement deposits account for most of the current and recent mine production. These pie charts showing percentage of contained antimony in US imports from the period of 2008 to 2011. The United States obtained 67% for its total imported antimony metals, ores, and concentrates, and oxidized from China. Resources and production. In 2013, the largest identified resources of antimony were located in Bolivia, China, Mexico, Russia, and South Africa. There are abundant identified antimony resources available, but the bulk of these resources are in a few very large deposits that are not evenly distributed across the globe. Antimony resources that may be mined in the future are likely to be those tied directly to deposits of precious metals, copper, lead, and zinc. Environmental considerations for the atmosphere. Global emission of antimony to the atmosphere average 6,100 metric tons per year, which include 3,500 metric tons per year from unsynthetic sources and 2,600 metric tons per year from natural sources. Water. The solubility of antimony in water at room temperature reaches 90 ppm in oxygenated waters, 13 ppm in oxide water, and 1.2 ppm in anoxic and sulfidic waters. And sediments. Published data shows sediments that range from less than 0.005 ppm in uncontaminated areas to up to 12,500 ppm in areas around a smelter. Plants. Antimony concentrations in plants do not show a strong correlations with soil concentrations. Plant concentrations range from less than 0.1 to 500 ppm. Environmental considerations. The mineralogical character of ores greatly influence the geochemistry of the mine waste. These three are the most common secondary antimony minerals in mineral deposits. The elements of great environmental concern are predominantly antimony and arsenic. In general, trivalent antimony is more toxic than pentavalent antimony. In humans, antimony can result in diseases of liver, respiratory, and cardiovascular systems and skin. WHO drinking water guideline is 20 ppb for antimony and 10 ppb for arsenic. From an environmental perspective, no clear case study of the behavior of antimony and related trace elements in a modern mine setting using current best practice exists. The toxicity of aqueous antimony species to aquatic organisms is a notable gap in knowledge. Knowledge of the toxicity of antimony in sediments is also limited. Thanks for your attention. That's all.
Thank you. Thank you very much for your brief report. It really widespread widened our perspective on antimony, considering its wide use in chemistry and industry. Well, now we're moving to Kwinizani Kwabe. Are you there?
Kwinizani, yes, hello. I'm here.
Okay, you can start. You can launch your presentation. 12 minutes.
Thank you. Is it sharing? Sorry. Okay, there you go. Thank you. Good day, everyone. My name is Kwinizani Kwabe, and I'm from the University of the Free State in South Africa. So what I'm presenting is still a work in progress in line with my PhD study, which basically seeks to devise a strategy for the promotion of indigenous vegetation. And all this is geared towards achieving agrobiodiversity management and food security. So agrobiodiversity, when broken down, is actually agricultural biodiversity, which includes a variety of indigenous flora and fauna, which are important components of food security, agriculture, and people's livelihoods. But my interest lies strongly on vegetative species, both indigenous and invasive vegetative species. So currently, numerous studies have stressed the importance of agricultural biodiversity to mankind as it encompasses sociocultural, socioeconomic, and environmental elements. For example, this would include your food and nutritional security, your pharmaceutical benefits, and also other elements which may, for example, include the conservation of agricultural genetic diversity. The current challenge, however, that we have here in South Africa in particular is that of agrobiodiversity loss. And I think some of you may agree with me when I say that this is a global pandemic which most countries seem to be facing currently. But much literature that exists on this phenomenon comes mostly from the Western countries and not so much from South Africa or even Africa for that matter, because I even went as far as looking at what other researchers from other African countries have published. And this challenge is seen both in places with cultivated agricultural landscapes and in the wild where there's been an overutilization of certain species without any efforts of conservation whatsoever. And an example of this would be our infamous indigenous Momordica fodder, which is used when people have certain kinds of ailments, like your flu, if you've got a stomach ache, for example. And also, some living organisms rely on such for their sustenance. But if you dig deep into literature, these are some of, if you dig deep into literature, these are some of the things that you could pick up. There are also the things that I picked up that most of our challenges are as a result of poor environmental management and agricultural interpretation. which have and continue to lead towards habitat loss and land degradation, as well as unsustainable productivity. And this is a major crisis, I would say, which in its very nature causes ecological imbalance. So the objective of the broad aim rather of this project is to devise a strategy for an effective practice of agrobiodiversity management. And to do this, I first had to explore the role of agrobiodiversity on environmental management and its impact on human beings. And because my previous research, where I looked at the socioeconomic value of indigenous vegetation, pointed me towards the management of indigenous flora, I also seek to promote and to highlight the role that is played by indigenous vegetation on the broader ecosystem. So as mentioned earlier, that this is an ongoing study. This presentation is anchored on previous research, meaning that a qualitative evidence synthesis was undertaken, which according to Grant and Booth is essential in identifying the most significant items in a given field and about a particular phenomenon. So from existing literature, different critical analyses were done, which resulted in a development of different themes, as you will see in the following slide. First of which is the loss of agrobiodiversity. So the study findings indicate that there's two parallel downfalls that are brought about by the loss of agrobiodiversity. And these include spatial and temporal variations in the natural environment. And examples of this would include biophysical diversity, which essentially affects our soils, water, our biomes, and to some extent plant productivity. And secondly, climatic variations such as the macro and mesoclimate cycles, which ultimately result to random trends of weather patterns. And these affect our habitats, they affect our natural environment. And then we also have these three, which I'm going to group together because I know I'm only given 12 minutes. So the critical analysis of literature again indicates that the development of the agroecosystems and the integration of modern farming systems are vital for agroecology and food security. So based on these findings, it's crucial to value and preserve the useful indigenous species of our native region. And more especially since the most recent research further postulates the importance of such use, particularly at a time where the world is experiencing global warming, which comes as a result of climate change. And therefore, to address the existing environmental challenges, there's a great need for different role players to devise strategies that are geared towards maintaining a balanced ecosystem, which is also what I'm channeling my efforts towards in my studies. And this drive, I believe, could be better achieved through inclusive agro-production systems, which not only value invasive plant species from the West, but also production systems that include the integration of both indigenous and exotic species in the farming system, as well as the use of sustainable and environmental friendly production methods. So I conclude by stating that the embrace of agrobiodiversity is a first step towards environmental management. It's also important for ecological succession between human beings and their natural environment. And it's also worth noting that in South Africa, there's a paucity of literature on the subject. So context-based primary studies to triangulate evidence in secondary research are much needed. Thank you very much. I would also like to acknowledge ASAF, the Academy of Science of South Africa, for this opportunity, as well as the DSI and my academic mentors. One of them is with us. Thank you very much.
Well, thank you very much, Kwenisani Kwabe. We had a lot of brief talks today. I hope that it was not because I made announcement about the timing. I hope that was not the case. But anyway, since you made these brief reports, we're in a good schedule, we're in a good timing, and we still have about 13 minutes to discuss all your reports. All speakers, please stay online, and whoever wants to ask the question, please send it to the group chat or raise their hand in the list of the participants of the Zoom. Now I can see that some of you have started the discussion, like Dmitry and Taisha Michelan. I see they're exchanging messages. Again, I'm encouraging everyone to ask the questions to the speakers. And if nobody is asking, I would like to ask my question to Swati Singh. Are you there? Swati Singh. Well, you know, it is especially interesting for me, particularly because I'm a chemist myself and study the photocatalysts. Well, thank you very much for the interesting report on the recycling the metallurgy wastes. One particular note is, would you like to consider changing from the UV light or UV lamp to photodiodes? They're cheaper, they're more energy efficient. This is what we're using here in Chelyabinsk.
Right. So actually, in Mumbai, Mumbai is just near Arabian Sea. So, like currently, it's raining here. So the problem is that the availability of solar light was not so consistent. So that's why we did not perform experiments with solar lamps. But probably yes in future. But I tried with xenon arc lamp, which actually depicts, which is actually it simulates solar radiation. So we tried our experiments with that. With that also we obtained similar results for the steel sludge drive catalyst.
Again, maybe you didn't hear me well, but did you try photodiodes? Light emitting diodes?
Not photodiodes.
You didn't?
Not diodes. I've tried with I've tried with xenon arc lamp, which is a solar simulator. So that is what we have tried as of now.
Okay, and another question: which region, UV region or visible region, make the phantom catalyst work? Like, what is the UV cut? What is the wavelength?
For my lamp, the wavelength was between 200 to 400 nanometer for the lamp that I was using. And it was a wide spectra lamp. It was not a specific wavelength that I used.
Okay, okay, all right. Got it, got it. Thank you. Any questions? Guys, we still have about 10 minutes to the coffee break. Dmitry Vladimirov, I have a questions for you. Are you there?
Yep.
Okay, Dmitry. Hello. I mean, I consider your report very relevant to Russia areas because you live in the Voronezh area, and here in Chelyabinsk region we still have a lot of steppes, and our agricultural species are really threatened with the invasive species. And considering establishing a huge mutual collaboration, especially in economic dimension, establishing this Silkway, what are the policies that has been established or being implemented to prevent the invasive species coming through the China or back to China from Russia? Are you aware of that, or do you know anything about it?
Well, both Russia and China have quarantine services. These are organizations that have to control any invasive species distribution. But examples like New Silk Road, it's very hard to understand how they will work. For me, the most interesting thing is to clarify how do invasive species, how they will use this migration corridor. And the second interesting corridor is just a project, we call it North-South Corridor. It's a joint project of Russia and India. And of course, regulation of invasive species migration is a crucial thing to organizations like quarantine services in Russia, in India, in China. And I also mentioned seaports of South Africa and Brazil. I know that there are lots of invasive species concentrate there, and they use roads and railway, outer roads and railway roads for the immigration. So for me, as a scientist, it's the most interesting to understand how will they migrate and how they are actually migrating.
Okay, all right. Obviously, yes, we are mostly concentrated on the fundamental knowledge. Yeah, I agree with that. Also, Pedro Brancaleon, are you there?
Yes.
Okay. Excellent report on the ecosystem restoration. I mean, it's not the area of my, competence. I'm not really aware of all mechanisms of the ecosystem restoration. But could you talk about a bit, in detail, how actually do you estimate or evaluate the restoration level or potential? And for us, for chemists, for biochemists, for those who work on the molecular level, in the general terms, how do you evaluate or estimate the ecosystem restoration?
Yeah, that's a good question because, as I said, like restoration can be implemented for achieving multiple objectives. So restoration success is always context dependent.
Mm-hmm.
So if you are targeting, for instance, the reduction of the concentration of a given molecule in the environment, this can be one of the measures of restoration success. And this has been done in some projects. While also at the molecular level, you can be focused on nutrient cycling or, some processes operating at the chemical level at the environment. So it all depends on which are your goals, but we can certainly use some more molecular or chemical analysis to evaluate restoration success.
Okay, thank you very much. Very, very, straightforward answer. I appreciate it. We see some activity in the chat, and I was not able to read it, but if there is any of you who would like to ask the question, please raise your hand, and you can ask so that you could ask it publicly. I see that. Yeah.
There are two for me, so I can take advantage and talk.
Yeah. Please go ahead.
So the first one of another Pedro from Brazil. So
Pedro, are you there?
Yes, I'm seeing him.
Yeah, sure. Nice presentation, Pedro. Nice to meet you.
Nice to meet you, Pedro. So yeah, we have recently worked with microbial communities, at least as a measure of the impacts from some restoration measures. So I have tried, for instance, different herbicides to control invasive species, and we have used the evaluation of soil microbes using metagenomics as an indicator of the impact of herbicides in the environment. But at the same time, I have just started a new research in partnership with Wim van der Putten from Wageningen University. So we are trying to track the more functional relationships between soil microbes and plants in restoration systems. But it's all new. Nobody have addressed it before. And another question, if I have approached restoration in dry lands. No, unfortunately, my research is mostly focused on rainforests and like humid forests and not in drier ecosystems. Though I work in savannas, like the Brazilian Cerrado, but the main problem there is not the drought, but especially fire, invasive species, and other problems, but not desertification. Thank you.
Okay, we can now move to another questions. I see Samak Aleng Moniani is asking Pedro about the effects of the global warming.
Yeah, so was the last answer.
Yeah. Oh, yeah. You already answered that. Okay. Dear Mr. Swati. Yaroslav Rybkin is asking Mrs. Swati about the volume of the reaction.
Yeah, I'm actually typing an answer. So
Oh, okay, all right. But still, you
answer
can
that.
ask so that everybody could see you, including me.
Yeah, so okay, so we used a solution in, so the reactor was such that the lamp was inside, and that was surrounded by a quartz film, so that the glass does not absorb light. And then there was the water solution. So we have not encountered UV absorption by water. Firstly, we used an aqueous solution, so the absorption, which is like if you compare the spectra of water absorption, it is far lesser if you compare it with the organic compound. Like I was using chlorophenol, so it has OH group, it has chloride group. So because of which it has a much higher absorption, it has much higher absorption as compared to water. So yes, but we have not actually reported it anywhere, but we believe that it is not that high as per the literature. What I've read is that it is not that high.
Yeah, laboratory scale, I assume. Fananani Ramolifo is asking Dmitry. Dmitry, can you see the question in the chat about the assessment and of biological control agents?
Yeah, I have already answered this question, but I can repeat for all auditory. I personally believe that biological methods are the best. They are not so risky as chemical methods of invasive species controlling, and not so wasteful as mechanical controlling. I personally, with my Chinese colleagues, now we're trying to find the biological agent to control Canis canadensis and Alternanthera phylloxeroides. In Russia, we have a quite good system from the Soviet Union of Institute of Biological Security of plants. For example, one of them is in the southern part of European Russia, and also we're collaborating with them, trying to find new agents to control invasive species.
Thank you, Dmitry. Now we're moving to coffee break. Everybody can go and take your time for a meanwhile, and please come back at 16:10 Moscow time. See you soon. Окей. Меня слышно, да? Отлично. Hello, welcome back to the Ecology session. It's getting really late in China. Let's not steal their sleeping time. Let's move forward to the Pedro Mirieles from Brazil. Pedro, the scene is yours. 12 minutes.
Hello, everyone. Can you see my screen?
Что у него со звуком? We can hear you. Go ahead.
Can
And
you
now
see
we
my
start
screen?
the presentation. Pedro.
Hello, everyone. It's a pleasure to be here. Good morning, good evening, and good afternoon. I am Pedro Mireles from the Federal University of Bahia, and I will talk a little bit about the microbial communities of a changing world. In this presentation, I will provide a general overview of the theme, and I will talk a little bit about my previous work and my ongoing projects and some future projects we are planning to do. So there is 1 trillion is the estimated number of microbial species. They can thrive in any environment that you can think about life is possible. They comprise the unicellular bacteria, archaea, and eukaryotes. They have a huge metabolic diversity potential, and they can provide many benefits to the nature and humankind. The abundance of the microbes is huge. It's estimated that 10 to the 30 cells in the world, and their biomass is greater than the animals, for instance. They also is responsible and provides several ecosystem services such as the primary production, decomposition, nutrient cycling, climate regulation, carbon storage, control of disease propagation, pollutant transformation, and they can be applied in the bio remediation initiatives. And of course, they are responsible for many aspects of well-being and survival of their hosts, animals and plants. For they are involved in the behavior of animals and the growth of plants, for instance. So how to access this diversity in the potential functions? One can use the metagenomics approach. It is just to make sure we are all in the same page. So the metagenomic is the DNA extraction from an environmental sample, and then this genomic DNA are fragmented and sequenced in high throughput sequencing machines, and the sequences are stored in the text files, goes through a bioinformatic pipeline so you can interpret this information, this molecular information, okay? So one can assemble these little small short reads into larger pieces that can be assembled. Eventually genomes can be assembled, and we can reconstruct metabolic pathways, okay? So we apply this methodology and this approach, integrating with macro organisms data to explore the influence of human impacts on several environments. And I will highlight some studies we performed in coral reefs. Okay, so coral reefs are, everybody knows, is Nemo's house. It's very diverse, beautiful, but they provide food for many people. Millions of people depend on the fisheries. They provide a lot of income with the tourism, and they provide protection from the coasts. Okay, but these environments, the corals, are the main building organisms from these kilometers of structural environments, like trees in the forests, okay? However, these environments are being treated by global and local impacts. Global impacts we can understand as their sea temperature rise and the acidification of the seawater. This causes massive coral bleaching and disease outbreaks and loss of coral cover. And we can understand the local impacts, mainly the load of nutrients such as sewage, and also the overfishing, which remove the fishes, and also the herbivores from the coral reefs, and the coral reefs goes through a phase shift, dominated by corals to dominated by algae. This photosynthetic algae exudates dissolved organic carbon in the seawater. This carbon feeds the fast-growing heterotrophic bacteria that can cause coral disease, and this keeps on a negative feedback loop. Okay, so this is the context we were interested in studying. So we went to the Abrolhos complex coral reef systems, which is the largest complex of coral reefs in Southern Atlantic. High endemism of corals and fish we can find in this place, and we investigated the effects of the marine protected areas in this marine park here, and in the fish biomass and coral cover, and also in the microbial communities. We found that, as expected, the protected areas had the higher fish biomass, lower flesh algae cover, higher coral cover, in a healthier environment. Okay, this reflected in the microbial community. So we found higher heterotrophic fast-growing bacteria in unprotected reefs. Okay, in high abundant pathogens in non-protected reefs. So this is another a contribution we gave in the marine protected areas, in the use of these ecosystems in a holistic approach. We then investigated the ecology of the mesophotic reefs from Vitória Trindade chain, which is this seamount chain, and this is Trinidad Island, just in front of Vitória town in Brazil, southern Brazil. Okay, we investigated the benthic community and fish community, and also the microbial community using by the means of the technical diving. And we find also a similar pattern. The environment dominated by turfy algae was dominated by highly fast-growing microbes, heterotrophic microbes. In these environments, the corals were had more disease, and the metagenomics of these corals reflected these shifts also. We then investigated another, we moved to the Pacific coral reefs, investigated a very unique coral reef from Japan in highly hydrodynamic regime, and using the same methodology, same approach, investigating all the community levels from the macro to the microbial communities. And we assess reefs highly impacted and not so impacted, as you can see. And these coral reefs were highly impacted because of the outbreak of the sea star, Acanthaster plant, that eats the corals, and the coral cover reduced a lot. So we investigated this during 3 years of research in this place, and the coral cover was very, very, very low in these highly impacted reefs. Okay, but the and the turf algae cover was very high in all reefs, including the high coral cover reefs. The bait community similarity and the fish community similarity, you can see the difference between the highly impacted reefs, these, and the bait community and the fish community, okay? But we cannot see the difference in the microbial communities. All seawater in these reefs were dominated by fast-growing potential pathogens, including coral pathogens, which raises our attention and awareness of the resilience of these reefs. They are subjected to damage, okay? So moving forward, we also investigated the microbial community diversity and potential function for hosts, the coral host, the mollusks, sea cucumbers, rhodolites from Abrolhos Bank, and also the seaweeds and many bacteria by genomic analysis and taxonomy, investigating the diversity of these unique places. And then I became more interested in the land use aspects and global change aspects. So we are developing projects addressing these issues. And the first one that I would like to share with you is this study we are performing here in Salvador, close to
1 minute.
Salvador, in my okay. We are investigating the shrimp farming activity in the mangrove sediment microbiomes and the mangrove forest sediments. So we have the impacted mangroves by the effluents of these shrimp farms, and these recolonization ponds is very nice to address the land use intensity of this activity. We generate massive data and assembled a lot of genomes, and we have interesting preliminary results. We are also investigating the oceanic edge, the mesoscale oceanic edges in Antarctic microbiomes. We are investigating these structures, oceanic structures that have influence even in land climate, okay? And we already collected the samples, and we are now preparing to sequence those samples. We then, so my next steps, I'm really interested in aquifers and climate change impacts in these aquifers. The reasons are because the human population are already facing water scarcity. The 40% of global population may live under absolute water scarcity in near future, and Brazil depends a lot of groundwater. And the aquifers microbiomes is very diverse, and they play critical roles in geochemical cycles, the microbiomes. So I'm really interested in working with these ecosystems. This work was not possible without the partnership of many friends and colleagues from national and international institutions, and funding from these agencies. Thank you very much.
Thank you, Pedro. Thank you very much for the talk on the invisible universe of microbes in the marine ecosystems. And now we're moving to Daria Tambasova. Daria, привет. Hello.
Hello, everyone. Take a moment. Okay, my name is Tambasova Daria. I'm from Ural Federal University, Russia. Now I speak some parts about my research work. The presence of environmental pollution with heavy metals is a serious problem in the modern world. Natural reservoirs are particularly affected by this. Cleaning reservoirs and mine waters from non-ferrous and heavy metals is a priority task of the ecologists. The issue is particularly acute in industrial region. The high content of the salt of heavy metal in water can lead to the accumulation in the tissues of living organisms. After consumption, this leads to a violation of metabolic process, central nervous system disease, and damage of vital human organs. Also, it causes the cancerogenic effects. Metals such as mercury, arsenic, chromium, zinc, and iron are particularly toxic. All these metals are found in both natural and artificial water case, causing, of course, irritable threat of public health. One way to resolve this problem is to perform water treatment from heavy metals by specific adsorbent. In our study, the natural mineral halloysite from Chelyabinsk region, Ural mountain, was used for this purpose. Halloysite is a natural nanoscale clay mineral with a tubular structure that is part of kaolin group and clay minerals. Due to their similar particle size and larger surface area, clay minerals have shown promising adsorption potential. A halloysite particle has an alumosilicate crystal structure rolled into a nanoscale tube with a monolayer of water between adjacent layer of alumosilicate. Its surface was modified with functional group for effective sorption of various metal ions. There are many functional groups and ways to graft them on inorganic surface. We used organosilanes such as glycidoxypropyl-trimethoxysilane, chlorpropyl-trimethoxysilane, and mercaptopropyl-trimethoxysilane. For surface modification of the halloysite nanotubes. The modified halloysite nanotubes were analyzed by infrared spectroscopy, electroparamagnetic resonance spectroscopy, and elementary analysis. The success of the selenization process depends on a number of characteristics, such as solvent nature, precursors concentration, reaction time, and temperature. The data obtained show the highest percentage of functionalization were 51% for glycidoxypropyl-trimethoxysilane, 94% for mercaptopropyl-trimethoxysilane, and 76% for chlorpropyl-trimethoxysilane. The best solvent is toluene, and the best reaction time is 4 hours. The modified halloysite nanotubes were analyzed by electroparamagnetic resonance spectroscopy. The acid base and electrostatic characteristics for modified materials we are studying using pH-sensitive nitroxyl radicals as spin pH probes. As can be seen from the above data, the APR titration curve for halloysite nanotubes number 2, it's with mercaptopropyl-trimethoxysilane, give a maximum shift about the calibration titration curve for the radical solution to the left. This silane was found to cause of most significant change of the acid-base properties for the surface of the halloysite. Scanning electron microscopy was chosen as a method for confirmation of the fact of surface modification. Figure 1 shown as unmodified halloysite surface, while the other 3 shown modified surface. The halloysite nanotubes number 2 sample proved the highest degree of silane functionalization that we confirmed by the previous data. The infrared spectroscopy shown the presence of very small peaks, as depicted in the figures. These bands represent vibrations of the Mendelian group, and the change of wave number scale may be due to the presence of neighboring electronegative atoms or groups. This indicates the toluene is the best solvent for the three types of silanes. These are band based on halloysite in the form of a fine powder packaged in briquettes made of permeable material. As an adsorbent that does not contain a leaf component, halloysite is resistant to the toxic effects of aggressive water environments saturated with heavy metals. Therefore, halloysite bricks can be collected by the dam that blocks the riverbed and start filtered the water. Then bricks with adsorbed metals are sent for regeneration. The metals adsorbed by the catalyst are removed from the surface of the modified halloysite in various ways. This is effective and inexpensive technology for water purification by extracting metals from natural and artificial waters. This slide shows the process of designing a custom-made, personalized, region-specific product for health of environment, combining both scientific work and innovation, as well as a production area. It is possible to create technologies which are attractive for large industrial holdings in order to restore the ecology of industrial region and improve the quality of life for population. These developments make it possible to reduce the concentration of non-ferrous and heavy metals for non-lethal concentration for plants, with the subsequent prospects in bioremediation using heavy metal accumulation plants. We have presented a custom-made product for a specific region for a healthy environment, which unique and environmentally friendly technology for water purification from non-ferrous and heavy metals. Thank you for your attention.
Thank you very much, Daria. Thank you for another great talk on from a fellow chemist, another great opportunity to collaborate with our researchers. And now we're moving to Arpita Dalal, India. Hello, Arpita. You may now launch your presentation.
Hello, am I
Hello.
audible? Yes.
Yes, you are.
Can you see my? Yeah,
Yes, we
can.
can.
You see my slide? Okay, great. So, hello everyone. A very good morning, good afternoon, and good evening to everybody. And I am Arpita Dalal from Assam University, India. And today I'm going to present a part of my PhD work on aquatic insects and its ecological importance, and most importantly, its potential as a biocontrol agents of mosquito larvae. Aquatic insects are a very good fish food and one of the major components of aquatic food wave. And it's also a very good bioindicator, indicating our quality of the water by their mere presence and absence. And they're also very good biocontrol agents of mosquito larvae that I'm going to show in the rest of my slides. They're also very good business partners because we can use mosquito to create many designs of jewelries. And some aquatic insects also can be used as in a forensic investigation as well. Now let's see how they can be used as a good bioindicator of water quality. They are also called as a biomonitoring agents. So their mere presence and absence of different families of aquatic insects, can be either tolerant or sensitive, and which ultimately indicate the quality of the water, either it's polluted or cleanliness of the water. So how it can be done? It can be done with the help of family level biotic indices. There are many family level biotic indices, protocols like BMWP, ASPT, Signal 2. What they did, they have assigned different scores to different different families of the aquatic insect based on their sensitivity and tolerance. So higher is their grades, higher is their sensitivity towards different organic pollutions, and lower is the grade is means the more tolerant they are to different pollutions. So all over, if we keep on counting the different biotic indices, so for example, for one particular aquatic bodies, if the signal scores comes at if less than 4, it shows that the system is severely polluted. If it is like within 4 to 5, the system is moderately polluted. If between 5 to 6, it is mildly polluted, and more than 6 is indicated the healthy habitat of the system. So through this method, we can really can do a rapid bioassessment test of any freshwater bodies just by mere checking their aquatic insects communities without further going into the chemical analysis of the water. So this is some of my PhD results that I'm showing here. What I have done, I have selected some study sites, basically the lentic ecosystems, which is standing water bodies like pond, lakes, that is floodplain lakes, oxbow lakes, or agricultural fields. And I have collected the aquatic insects, and I have also calculated after that there are different biotic indices like signal, BMW, PNS, PAT. And from that, it indicated that most of the sites that I have selected for my study area is either moderately polluted or severely polluted in nature. To support this results, what I have done is I have estimated different chemical analysis of water. For example, free carbon dioxide, dissolved oxygen, nitrate, phosphate, total alkalinity, electrical conductivity, etc. And what I have found through CCA triplot, that is a canonical correspondence analysis triplot, that most of the species are positively associated with the water variables that are totally indicating the pollution condition of the system. And some species are also found isolated from the environmental variables of water, which indicated that these species are opportunistic species and they can thrive in any conditions of the water quality. So these are some of the aquatic insects that I have recorded during my study site, and all the photographs are taken by me under the compound microscopes. And I have recorded a total of 99 species of aquatic insect during my 2-year study in my area. This is some of the publications that I have made from my biomonitoring work. Now comes their role as a biocontrol agent. So there's a chemical way of controlling mosquito has many side effects, and we have to control the mosquito because it is a vector of many deadly diseases like malaria, dengue, filaria, chikungunya, and whatnot. So there are many biological control agents are already in the environment, right from the microbes to zooplanktons to fishes like gambusa, guppies, etc. Apart from that, aquatic insects have the equal potential to control the mosquito population, but less study has been done checking the aquatic insects as a predator. So biological control study in a laboratory condition can be done with the help of functional response as well as multiple predator effect. Now, what is functional response model? It is a prey density dependent model which comes up in 3 different shapes, that is type 1, type 2, and type 3. In type 1, as the prey density increases, the predatory rate keep on increasing linearly. In type 2 model, even at the low prey density, the predatory rate is too high, and as the prey density increases, the predation rate keeps on increasing till it reaches asymptote. And in a biological control study, type 2 functional response is quite desirable because it leads to prey population destabilizing effect. Now comes type 3 functional response, which is a sort of is more like a learning behavior, because here at the initial prey densities, the predator is trying to learn how to catch or subjugate the prey, and as the prey density increases, their predation rate keeps on increasing. So I have done my functional response experiment first phase at Queen's University in UK, and the second phase at my home university, that is at Assam University. So I have taken some focal predators that is available at the temperate zone, that is Notonecta and Glauca, and Gammarus pulex. And for prey, I have taken Culex pipiens. I have taken all 4 larval instars of the Culex species. And at India, I have taken again 2 different predator that is locally available, and this predator is quite unique in nature because it don't chew the prey. In fact, it has a unique mouth part like a needle which pierces the body part of the prey and suck the body fluid out of it. So for the experiment at Queen's, I have taken only one kind of container size, but at the work that I have done in India, I have taken 9 different type of container sizes divided into 3 different water depth and 3 different surface areas, just to see how the predatory behavior is changing depending on different container sizes, because we are more interested to control the mosquito that are container breeder, that like Culex or Aedes. Now comes the experimental design. Both functional response and multiple predator effect has the same experimental design, but in multiple predator effect, we use more than one predator per arena. Now comes this is about how the experimental look likes. We use a 24-hour starved predator and offer them the different prey density of 2, 4, 8, 16, 25, 50, 75, 100, and the different larval instar were given separately. And same set of experimental trials are also been there, but it is without the predator to see how if the mortality of the prey is solely due to the predation and not some other factors associated with it. Now comes, this is the experimental setup at Queen's. This is the experimental setup at Assam University. Now comes the result. We have seen that this is a result from the Queen's. We have seen that both the predators are good at predating all the 4 larval sizes of mosquitoes, but second instar larvae was most fed throughout the study period. And it has been seen that Nodonecta glauca is far more ferocious predator in predating the mosquito larvae as compared to the Gammarus pulex. Now comes their multiple predator effect. Here we have used the conspecific pairs, not the heterospecific pair, because of some hindrances. Here we have seen that conspecific pairs of a Nodonecta glauca is far more better than as compared to the Gammarus pulex, because Nodonecta glauca is showing some synergistic behavior with each other, leading to more prey than expected they're eating. Whereas in case of a Gammarus pulex, their behavior is more like an antagonistic behavior, which led to less number of predation as expected. Now comes the work that I have done at India. Here I have seen some sort of variation in the overall predatory behavior throughout the different sizes of arena, but overall both the predators are predating all the 4 different sizes of prey larvae. Out of the second instar larvae is again most fed one, but out of that we have seen that Anisops sardius is the much more better predator as compared to the Anisops brittonii. And now comes their multiple predator effect. I have taken 3 different combinations of predator, like on their conspecific pair as well as their heterospecific combination of both the predator. And we have seen that again conspecific pair of sardius, Anisops sardius, is better performing as compared to the rest 2 combination pair, because it has showing less antagonistic behavior with each other as compared to the rest. And we have again also seen that while predating the low size prey like first and second instar prey, they have shown some kind of antagonistic behavior, because they are more interested to catch the low size prey. But as the prey sizes increases, when they reaches the 2, 3rd and 4th instar of prey sizes, their overall antagonism behavior keeps on decreasing, because they are not interested to catch the larger size prey. So this is about the heterospecific pair I have already discussed. And this is the publication from my pie control work. From my work, it is clearly seen that aquatic insects has a very good potential to control the mosquito larvae. Both the aquatic insect present in the temperate and tropical region have the equal potential to control the mosquito larvae. Basically, the container breeding mosquito that lay eggs in the backyard of our house. So I'd like to thank my collaborator from UK and France, and thanks to my funding agencies. Thank you very much. And the take-home message from my work is biological control is the future. Thank you.
Thank you very much, Arpita. That was really intense. Let us all acknowledge Arpita's brevity as she was working with the really, really spooky insects. I also really like the idea of the bioindicators, because it's always great to substitute the complex and fragile electronics with the funny bugs. We're now moving to Dongmei Yang from China. Dongmei Yang.
Hello?
Yes, you may now start the
okay.
presentation.
Can you see my PowerPoint? Can you see my PowerPoint?
We can see you, but please share the screen.
Oh, sorry. I didn't share that. Okay.
Everything looks fine. Go ahead.
Okay. Hello, everyone. I'm Dongming Yang, come from China, Zhejiang Normal University. Today I will give a presentation about water extraction method for estimating the resistance to cavitation in the long vessel species. We all know that water is very important for the growth and the distribution of species. And during the water transport from the soil to the leaf, a cohesion tension theory is very important. We can see water evaporation from the cell walls of the leaf cells generate large negative pressure, and this negative pressure transmitted to the xylem, and then they pour water through the xylem. In the whole process involves the movement of tensile water that is metastable. Metastable, that means the water is unstable. So the water column in the xylem is easily break down. That will result in cavitation and embolism. Cavitation, that means the gas bubble or the air go into the vessels or tracheas, make the functional vessel become the dysfunctional, become embolized. That means the air replaces the water in the tracheas or the vessels. This will prevent the water move in the plant, so that will reduce the hydraulic conductivity, pH. But the species or plant can evolve special structures. We can see there's lots of small pores in the pit membrane that will prevent embolism from spreading between xylem conduits. That means if in a long vessel, it is cut open without end, that means without pit membrane. At this case, the nanoparticles or the bubbles will be very easily go into the vessels to induce cavitation. Scientists use the vulnerability curve to estimate the resistance to cavitation, and this is the curve. Uses the percentage of cavitation or percentage loss of hydraulic conductance, PLC, versus xylem pressure. We can see the two types of typical curves. R shape, that is represent the low resistance of species, because just a very small negative will have a big cavitation happened. But S shape, that means the species is a high resistance. And when the PLC equal to 50, corresponding to the xylem pressure, it's called P50 values or T50 values that can be represent the resistance to cavitation. Until now, there are more than 1,000 species have been reported about the P50 values to explain how about the species draft tolerance. And this picture is about the cavitation happened in the leaf vein. So we can see the cavitation resistance is very important in ecology and physiology. There are different methods to measure the vulnerability curves and the P50s. Most frequently used is the benchtop dehydration. It is the oldest, and scientists think it is gold standard. That means if the new method used must be compared to this method to see if this new method is correct or not. But this method is very, very slow. Normally we need 2 or 3 weeks to finish one species vulnerability curves. And another is the centrifuge method. It is widely used because it's fast. However, it also has some problem. There has been a lot of debate in recent years, especially for the same species, especially for the long vessel species. We use different methods, can get different curves, different P50. For example, in Robinian, if we use centrifuge method, we can get a typical R shape. If you use benchtop dehydration method, we get a typical S shape, and the P50 can range about 3 MPa. So how about this species property? This resistance or not resistance? Then if we measure the water potential in the whole day and the gas conductivity, we can find that the Robinian is S-shape behavior should be S-shape vulnerability curve. So the centrifuge has some problem. So today I will talk about centrifuge method, how to improve it. In fact, there are 2 types of rotors, Berry rotor and Crochet rotor. In fact, this is the 2 researchers' name, and they have different principle, but I will not detail talk about that. I want to focus on that Crochet rotor. They use 2 cuvettes here, and this is the stem. The 2 cuvettes have holes here and here. This hole is high and this low. So during the experiment, the water can flow from the high to the low level. And in fact, in both methods, the water can move into the stem. If the vessel is very long, and that means it's cut open. It's cut open, just like what I said, without the pit membrane to prevent nanoparticles. In this case, the nanoparticles or the bubbles can go into the stem. This will induce the cavitation, but this cavitation is not a natural cavitation. In fact, it's an artifact. So we call this open vessel artifact, that because nanoparticles go into the stem during experiment. So how to avoid this nanoparticle enter into vessels? The solution is a water extraction method. That means the water. That means extraction will push the nanoparticles out of them instead of going into it. This idea is not new, first reported in 2013, but at that time it's not precise. And most important thing is they didn't consider about evaporation during the experiment. So we improved this method. We use the 2 cuvettes without hose, and the two water level the same. We consider the evaporation and the extraction. When the rotor is spin, the water can extract it from center to the end, and we based on the water level and the all water extracted volume to represent the cavitation. But must make sure the water is extracted from the vessels or xylem, not from non-vessels, because the boundary curve is trying to say about the xylem hydraulic conductivity. A typical water extraction curve is like this one. In the beginning, the extracted water volume increase, and then when the evaporation rate equal to the extraction rate, it will reach the peak. Follow this, evaporation dominate. In the first report, they think that the peak value is the extracted water volume, but we know they didn't consider about evaporation, so it's wrong. Firstly, we from theory to prove how to get the correct extracted volume. This green is evaporation without any extraction, and then this one is the theoretical extraction without evaporation. And in fact, what we can see in the experiment is the combined impact of extraction and evaporation. So we, from the theory, we prove that this intercept always equal to the maximum extracted volume. So from here, we can say the first report, they are underestimated the total water extraction. Then we did the experiment in Albania. Firstly, we used the dye to use the material to dye the vessels. If the vessel is functional, that means we can stain it. Otherwise, if they are captured, it will cannot be stained. So from this primary experiment, we proved that the two phases, there's a percentage loss of water volume. The second phase is the water extracted from vessels. And the first phase, the water is not extracted from the vessels. We just say the extracted water is non-vessels. So we only focus on the second part to see how about percentage loss of water volume can represent the percentage loss of conductivity. Then this is what we can do. We can get the peak value. Also, we can deduce the maximum extracted water. We repeat 6 samples, and we can see that the percentage loss of water volume is very identical. And from our improved method, we get the extracted water volume is 2 times our peak method. And also, we did an anatomical analysis. We found our extracted water volume is equal to the volume of embolized vessels. So what we did is correct from here, and then we use the Weber curve to fit the percentage of loss of volume curve. We get the RMS in our method is significantly lower than the peak method. So our method is more precise than peak method. However, the percentage loss of water volume is not equal to percentage loss of conductivity, because volume is not mean the conductivity. Although cavitation, we can see air replaced the water. Then we based on some theory and assumption, we got the correlation between PLC and PLV. We call this PLC theory, and then we plot this PLC. We can see theory PLC, this red curve, compared to the benchtop dehydration, this green curve. This is, we said this is the gold standard, and we found the T50, there's no significant difference between our method and the gold method. So we can get the conclusion. Firstly, our improved method can produce a better estimate of the total water extracted volume and the Weber curves. And then our method deduces the theoretical PLC, the P50 or T50 is no different to the benchtop dehydration. Also, it is consistent with the water relations of Albania. From here, we can say it's maybe somebody think it's just a method improved. How about what's if it's very important? Yes, I think I want to say it's very important, because in the past many years, there are also reports about vulnerability curves for long vessel species. Most people report this is R-shaped. It's purely for traditional centrifuge method. This is not really R-shaped because their method is wrong. So we have lots of work needed to do. In future, we should check these species to see how about their properties, if they are really resistant to cavitation or not. So this is why it's very important, because we only can get correct method, and then we can really understand this species property. Then we can understand the distribution pattern, or why the species can adaptation in the special environment or competition. Also, somebody has proposed the complicated correlation between resistance to cavitation, conductivity efficiency, and the mechanical strength. So I think in future, we can do lots for this in ecology. That's what I did. And I wish in future I can get some cooperation with you. Thank you.
Well, that was really interesting. Thank you very much, Dong-Mei Yang. That was really excellent report on the novel estimation of the cavitation effects. I really do hope everybody enjoyed that. And now we're moving to Semakaleng Monyai from South Africa. Semakaleng. Hello again.
Hello. Hi.
Hello. You now may start, and you now may launch your presentation. Yes, we can see you.
Can you guys see my slide? Okay.
Yeah, start sharing.
Can you see my presentation? Okay, thank you. Hi everyone, my name is Makaleng Munyai from the University of Pretoria, South Africa. So I'll be giving you a talk today on the exploring of microbial.
Semakaleng, we cannot see your presentation. Please share the screen.
Oh, okay. Okay, sorry about that.
No worries. Go ahead.
Okay, so I'll be giving you a talk on my project called the mining of environmental DNA polymerase, which are novel from South African side. So basically, I give you a background on the environmental methodology. So natural resources such as soil, deserts, and geothermal vents and hot springs have been known to have a high diversity of microbial communities. These communities, they are actually having a high protein similarity, but they wish to show to have like dissimilarities, variations within the amino acid sequences. So extreme soils serve as a reservoir for microbial communities, for microbes of microbes which have a high source of thermophilic enzymes. So they are actually targeted for bioprospecting because of their exploitation in different kinds of biotechnological applications. This is because they have different properties and activities. And just to mention a few examples of those enzymes is bacterial amylases, lipases, proteases, and nucleases, and polymerases, of which will be of interest to my study. This is mainly because they are involved in polymerizing the nucleotides during DNA and RNA synthesis, and are used in different sequencing platforms. So DNA polymerases are classified into 8 families. Then I'm going to be focusing for this presentation on the family A. So just to show a structural depiction of how the domains are organized. So they actually have the 2 bidirectional exonuclease domains. And then they also have a DNA polymerase domain, which is the most active site of these DNA polymerases. And that's just to show you the 3D structure of this pose. So ideally, they are mentioned to have like a hand, a shape of a human hand, the right hand, and with the finger, the palm, and the thumbs, and the polymerase active site is actually located in the palm domain. So just to look into some properties that that actually make them to be considered essential is because they are actually involved in, for example, the finger in substrate specificity, where it actually contacts the incoming nucleotide and serves as a binding site for the template DNA. And the thumb is actually involved in sensitivity, and which actually guides the formation of the newly formed DNA. And the most important one is the palm domain, which is the catalytic active site, as I've mentioned before, that is involved in binding of the template DNA and contacts the 3' group of the primer strand. This actually helps to keep the DNA in place when DNA synthesis is happening, as you can see in this figure. So they are used in different kinds of biotechnological applications as I've mentioned before. So this is just to show a few. But then of interest for the study is to look for DNA polymerases that are being used for next generation sequences, because we have different sorts of platforms that are used for sequencing. So different types of DNA polymerases are applied for depending on the requirements of the sequencing platform. So to look at why am I looking at soil, basically, it's because a study in 2016 actually profiled the metagenomic study at a broader scale, where, as you can see in this figure, most of the metagenomic studies, sequence-based analysis, is actually focusing on just checking the microbial diversity of microbes in different environments and ecosystems. However, the studies that are looking for enzymes, there are quite a few, as you can see with the red dots on the figure. So South Africa is actually known to have a variety of soil ecosystems. And then this may serve as a potential source of novel DNA or novel enzymes in general. So then we are looking at this because we are trying to look at the discovery of these enzymes from this soil, because we do have some kind of extreme soil environment. So the project aims to identify, as I've mentioned, so we are going to use sequence-based metagenomic approaches for the study. So then how was this done? So based on the previous map that I showed of the South African soil different soil metagenomes, so 16 samples were collected, and DNA was recovered from each of these samples in 3 different areas, which is the arid, semi-arid, and the hyper-arid. And then DNA was extracted from that, as Pedro was mentioning earlier about the sequencing platforms. So it was actually extracted and sent into for sequencing, and the data set was actually analyzed using different computational tools. So then the first thing we did after getting the sequencing data is I actually did some data processing where we do quality control and trimming to actually get rid of the bad sequences. And then we actually get to assemble those tiny sequences to try and generate the reference sequence. So then this is what we saw from our data set. So we analyzed about 30 gigabases of data, of which the raw sequences were like 250 million. And then after the trimming and the quality control, in total, from the 4 samples, we ended up with 140 million sequences, which we actually proceeded with for downstream analysis. So once we have those sequences, we actually did some DNA polymerase prediction for the ORFs that are coding for that. So, and also did some taxonomic annotation, and we also used curated databases to actually verify the ORFs that we actually identify and predicted from our metagenomics. So then, in order to do this, we used Cytoscape to actually get like a whole list of the genes that we managed to find, and then we actually annotated each cluster separately to try and see how are they clustering. So for this study, because I'm actually looking at DNA polymerases, then we managed to find a different set of polymerases in this particular form. But then, since I'm interested in DNA polymerase 1 for the interest of this presentation, we managed to identify 16 DNA polymerase genes from this particular one metagenome, of which 6 of them were actually complete ORFs that had the 2 bidirectional exonuclease domain, the polymerase domain, and then in total, from the 4 metagenomes, we ended up annotating 75 genes of the DNA polymerase A. So now, once we've gotten all this, we actually combined the sequences from all the different types of soil samples, and then we actually constructed a phylogenetic analysis, where we actually classified them based on, before we did that, we actually filtered them because we had some that were mesophilic and some that were not related to any thermophilic environment. But this is because we are looking at extreme environments. So then we filtered all the sequences, and the sequences that we actually did a phylogenetic analysis with, and we are going to continue working with going onwards, are those that are actually thermophilic. So as you can see on the tree, we actually have like thermophilic ones, hypothermophilic, and the ones that are clustering based on UV radiation, and the commercialized ones. So we identified a range of sequences that were between 37% and 80% when we compared them with the curated databases. And this we're looking at thermophilic organisms, as I've mentioned. And then some of them did cluster very closely with the characterized polymerases. And however, some of them didn't even actually get a cluster with any of them. For example, the cluster that you are seeing here, they're actually clustering alone. And none of them clustered with TAG or Ecology actually at the same branch. So then we went on to do some functional evolutionary conserved motif analysis. So this is how this was focused on the polymerase domain. So basically the polymerase domain is actually classified into 6 different motifs, which are actually involved in the polymerization. So then now the polymerase activity is actually mainly targeted to the palm domain, because that's where the active site is, as I've shown earlier. So then we try and see the conservation of the motifs, and we saw that they actually conserved very well with a high conservation. And then most studies that are actually looking at the polymerase activities are focusing on the motif A, B, and C, which is what we are going to actually look into as well in the nearest future. So then on those conserved motifs, we actually went on now deep to check in our sequences, how many polymorphisms are we actually seeing and what do the changes mean, basically. So then we only managed to find that, yes, we are seeing a lot of changes within our polymorphisms, within our sequences that are different from the canonical ones. But then now in terms of function, what does this mean? So, and how does it actually address the properties and the functions of this pulse? So then what we ended up doing is we went into literature to look into some of the mutational studies that have been done, and that have been studies that have characterized different site-directed mutagenicity of these motifs. And then we only managed to find that the blue ones, as I mentioned, they're the ones that are actually have what type activities, and some of them, they are actually involved in chain termination, infidelity, in substrate discrimination, and specificity. So then we, at the end of the day, we managed to see that we have a high diversity of poles in our soil systems. We also went on to look at other sequences that are other residues within the sequence that are like in close proximity to those active sites. So we managed to find that there's quite a couple that are actually involved in primer extension rate, which is actually higher and comparable to TAG, for example, in row number 1, and we got 10 sequences that have that substitution. We also saw that there's high displacement, which is actually involved in polymerase activity. We have 22 samples that are involved in that. And then if you delete the last part of the C of the polymerase domain, you actually find that you reduce processivity, and altogether the enzyme doesn't actually get to function. And we managed to see that we have 9 sequences from our metagenomes that are actually like that. So we have managed to identify 75 novel DNA polymerases, and then we from this 37 of them were actually related to thermophilic organisms, and we infer that as we have identified this, they may have the same function as polymerases due to the conservation of the essential residues. So the metagenomic polymorphisms of these residues, that's what we're actually trying to explore. So then the results actually demonstrate that the soil samples usually contain a high level of genetic and enzyme novelty. And then this suggests that novel thermostable DNA polymerases with new properties and activity for the potential application in various sequencing platforms. As I've mentioned, that's the significance of why I'm doing the study. So the future work since now we've managed to see like some few that have like potential interest in sequencing, we are going to actually send this for gene synthesis, as well as at the same time in parallel, we're actually going to actually clone this from the metagenomes and try to compare this, actually try to express them and purify in the lab and use this for some characterizational assays later on in the lab. So I would like to acknowledge the following: my supervisors, the lab mates, some of my mentors, and my funding bodies, and my institution for the enrollment. And then thank you.
Thank you, Simakaleng. This is a really great job on screening of huge amount of microbial genomes. Great job. Thank you very much. And now we're moving to the questions and discussion of the on the reports. I have seen there were a few questions in the chat. For example, Yaroslav Rybkin was asking Daria about the recovering the sorbents. Daria, you ready to answer that question publicly?
Yeah, thank you, Yaroslav, about your high interest about my research work. Well, I start with the first questions about halloazite works depending of 3 days to 7 days. It's because water pollution is different. Sometimes we have a high degree of salts of high metals in water. Sometimes it's low. But we have special reactions with special city. You know some about Chelyabinsk region is a city of Karabakh. City of Karabakh have a river, and we cleaned this river. We completed this 3 days, and after that we start to removing or sorbents. Sorbents we remove different ways. For example, it's electrolysis for metals and chemists reactions. This sorbent not specified because this sorbent added to all metals solving in water. And after that, we remove these metals for different way with electrolysis. I think it's enough.
Your question. Dongmei Yang, I have a question for you. You did a great job on developing a new method for estimating the cavity effects. Dongmei, you hear me?
Hi, yes. Yeah.
Yeah, the question is, where else except the, the area of plants, the pores in plants, where else could your invention be used? I assume that these developments has some application somewhere else, not only in the plants.
In fact, this method is, the cavitation, we said this cavitation just the air happened in the xylem, so it's only for plant. It can be the trees, also can be other shrubs. But what I used to improve the method is for long vessel species. Long vessels, normally long vessel species will be distributed into the, not very cold, because the cold area always the short vessels. So for the tropical area, this part species I think it's the most is the long vessel species or some wine. So it's also long vessel species. So if researchers want to do this, they should be careful, use the traditional method. So only can use the implant, not others.
Okay.
I don't know if what I answer is okay.
Yeah, that's fine. Semakalong, I really liked your report on the screening of the microbial genomes, and my question is, Semakalong, are you there?
Yes, I can hear you.
Yeah, I mean, it's a great job. I see you screened a lot of information, and you specifically have extracted these 75 genomes and the very potential of the very effective polymerases. So on which particular organisms these polymerases are applicable? Like what kind of genotype do they polymerize? Microbial or higher states organisms? Is it applicable only for microbes or anything else?
So it's a wide scale application.
Wide
so
scale application.
You can, so you know, when we do sequencing, for example, if you remember Pedro's talk, he mentioned that they got samples from the ocean, from the coral reefs, and then they actually did sequencing and stuff like that. So basically, when we get these enzymes, we're actually trying to get something that is like similar to what's there or that has more advanced activity as compared to what's used in PegBio or what's used in nanopore, what's used in different platforms of next generation sequencing as well in Senga. So it's basically an enzyme and new novel enzymes that will be used at a broader scale, which will be having enhanced activity.
Okay.
So they actually amplify everything. They don't have
it.
like a specific pattern.
Okay, I got it. Another question is, semicolon, is what it had to do with the thermophilic microbes? Why thermophilicity was crucial in your research? You mentioned the thermophilic microbes. Why them?
Yeah. So it's mainly because most of the industries and biotechnological applications, they do processing and they actually have like processes that are like at high temperatures, for example. So then if you just get an enzyme that is not thermostable, basically it's not going to be functional for the biotechnological application. But then if you get an enzyme that is actually, okay, let me just say any biocatalyst that is actually thermostable, that means it can actually function in terms of any temperature whatsoever. But then in this case, because now when we look at sequencing, remember there's a step of most of the PCRs is actually from 65, 64, 58 up to like 98 degrees. So we expect that it should have the same thermotolerance, because if you're using for next generation sequencing, for example, you would actually expose it to high temperature, so it must be able to actually sustain and function in that high temperature, for example. So if you find, I don't know, does that answer you?
Yeah, it does. It does. Thank you.
Yeah.
Also, dear participants of our session, I strongly encourage you to send the questions. There were a really excellent reports today, and I'm sure you have a lot of questions to ask. Don't make me ask boring chemical questions. Please ask some, diversified ecological questions. Okay. Also, my technicians are sending me the message that tomorrow's speakers, please, we're reminding you that you should appear 1 hour prior to your presentation to make sure that your presentation can be viewed and you have no problems demonstrating it or don't have any problems with the sound. I still don't see any questions appearing. That is really strange. I really enjoyed the microbiome reports of Pedro Mirieles. I mean, he did a great job. He did a great field job and developments. If I were able to ask the questions, I would really love to ask, but I'm not competent in that. Now we have a question from, okay, that's not a question. It's just saying thank you. Another great report from Arpita Dalal. Again, I'm not really good at the bioindicators, but the whole idea of substituting the indicators, the regular indicators with the funny bugs, is just phenomenal. I would really love to, if someone would spread or develop this idea in the discussion. Okay, Thaisa Michelin is asking Arpita. Thaisa Michelin, are you ready to ask the question publicly?
Hello.
Hello, yes. Go ahead.
Yeah. Thank you, Michelin, for the question that you have asked. Actually, BMWP and ASPT is already a well-established protocol developed by the European countries, and then in Australia, and then some part as some modified part of BMWP and ASPT has been developed in Thailand and some Asian parts. And such protocols, you can use again and again when you're going to do some kind of biomonitoring work on rapid bioassessment work. So yeah, it has been applied in India before, just taking the well-established protocol that has been developed in Europe and Asian countries before. So we have just taken that and used in our present work. And our adaptation is not particularly the European work. We have taken the adaptation of BMWP and ASPT from the work that has been done in Thailand, because it is more related to India, because we are from the same Asian country, so it has a higher probability to get similar kind of aquatic insects from Thailand as well as from India. So I hope I can able to answer to Michelin.
Okay, we have a response from Thaisa Michelin. Thank you very much. Any other questions? You still can raise your hand in the list of the participants. Okay, we have another question. Again to Arpita Dalal. Arpita, I hope you see the question.
Yes. Yeah. I see this index is used for family level value. How do you account for sensitivity difference with single family? It's a the sensitivity or tolerance difference has already been done by the expert workers in this field in Europe and Asia and in Australia. And the sensitivity they have done based on their tolerance level to different kind of pollution. If some species are very much sensitive towards like Ephemeroptera, Plecoptera, and Trichoptera, EPT is one such group which is very much sensitive to very different kind of organic and toxic pollution. That's why they cannot be found in any toxic or polluted water bodies. They are always found in a stream, higher altitudinal stream where the water is very much clear. You will find always EPT over there. So in this way, the sensitivity grade has been assigned. And some species you'll always find in a very much dirt and low quality water, like chironomids larvae and then mosquito larvae. So their sensitivity grade has been assigned by the previous workers, not by me. I have not done the BMWP and ASPT protocols. I have just used their protocols in my work. Thank you.
Thank you, Arpita. Thank you again. Again, we still have a few minutes left for your questions to be asked. I gotta mention today was a really nice and organized reports. Everybody stayed on the great timing. I appreciate you for that. Hope we will continue in the same regime tomorrow. Mm-hmm. Screening. Okay, we have another question. Just on the thermophilic questions. Yeah, thank you, Samakalang, on the extending the response. Mm-hmm. Yeah. Roger that. Okay, I think that sums up our today's session. Thank you very much for participation, giving us a great talks, and I hope to see you tomorrow. Have a great day, have a great night, wherever you are. See you tomorrow, guys.